Hi everyone, how are you? So, welcome to another video of inheritance and variation. We are on chapter 3 for the Maharashtra board and chapter 4 of CBSE. So, let's begin. In this video, we are going to see the chromosomal theory of inheritance. We are going to see about the chromosome structure in a bit detail and the two types of sex chromosome, that is X and Y, that also we will be discussing in detail. Okay, so we have finished the first half of the chapter that was discussing about Mendelian genetics and neo-Mendelian genetics, that is deviations from the Mendelian genetics. In that we saw 4 different categories: incomplete dominance, codominance, pleiotropy, multiple allelism. Okay, now we move to another part, a shift from the Mendelian genetics. Here we will be discussing the theory of how chromosome, it was discovered, and what is the structure, what is the function, and what are the two different types of sex chromosome, how do they look like, do they have any role in sex determination and stuff like that. So, this theory of chromosome was proposed by Walter Sippel and Theodore Boveri in 1942. Okay, Theodore Boveri observed that proper embryonic development of this animal sea urchins does not occur unless chromosomes are present. So from that, it was known that chromosomes are some of the basic entities required for development of life. Okay, without DNA, now we know that the genetic material is DNA, chromosome. Without chromosome, there is no life, and that's the actual molecule entity which is required for the life and development of the life. But earlier, it was all these things were not known, and that is how research and science came up. And now we are aware of so many things. So Theodore Boveri was the one who discovered this, observed this. Then Walter Sippel observed the separation of chromosome into daughter cells during meiosis. Now we know, okay, there are two types of cell division. One is mitotic, one is meiosis. But when genetic recombination happens, that is exchange of chromosome between the father-mother chromosome is taking place during meiotic division, then there are a combination of new genes which are formed and new traits, and combination of the father and the mother, genes takes place, and that is how you can see some of the traits of the father and the mother in the child. So now we are aware, earlier it was known, so things were just getting discovered. So Walter Sippel observed that, okay, the chromosome has to separate, they has to come together, and during the meiotic cell division which happens in germline cells. Okay, so what is the general understanding of this theory is that pairing and separation takes place of the chromosome. It says that pairing and separation of a pair of chromosome will lead to segregation of a pair of factors that they are carrying. Okay, just like you can see in the representation over here, one set of parents over here, grandparents, one set of grandparents over here. From their children, okay. So first, so they came together, and that is how their genetic recombination brought about their, they got their children. So pairing, separation. This is how generation by generations are formed. Okay, so genes are getting transferred from one generation to another. Along with the transfer of genes, you will see certain similarities and you will also see certain differences. So similarities and differences will come hand in hand. Okay, so some of the important statements you should know is that what is getting transferred, how are the genes getting transferred from the parents to the child, to the offspring, via chromosome. Chromosome basically is made up of DNA which carry the genes. Okay, they are transmitted from parents to offspring. Two identical chromosomes with similar genes are homologous. These you have studied in the lower standard, so nothing new in this. When there are certain regions of the chromosome which are having similar set of genes. So let's just consider, for example, just imagine that this region and this region, for example, are having same set of genes. So this part of the blue chromosome and this part of the red chromosome, we will call them as homologous pair. Homologous means homo means similar. So two identical chromosomes with similar genes, similarity between them, they will form a homologous pair. Okay. Now let's see over here. Let's say for example, these two are homologous pair. These two are homologous pair. They are known as sister chromatids. Okay. Fine. So chromosome are mutable. That is, mutation can happen in them. Certain defect, certain disorder, so change can happen in the chromosome. So it will not remain the same always. Mutation can happen to them. And genes are present on the chromosomes. Chromosome is made up of nothing but deoxyribonucleic acid, that is your DNA. And on this chromosome, on this DNA is your genes, which are transmitted from parents to offspring. Chromosome will act as a vehicle of heredity. Understood? Homologous chromosome and opposite of homologous will be heterologous, dissimilar genes. Fine? So hence they show similar behaviors. Fine? So genes can be transmitted from parents to the offspring. There will be similarity as well as there will be differences. Okay. Then this chromosomal theory of inheritance more or less is resembling the Mendel's law, and because of these following observation, one observation was homologous pairs are independent of other chromosome pairs. So homologous pairs will, the trait which is coming from the homologous pair, the gene which is coming from this region of the chromosome, will be expressed independently, as we saw in the second law of third law of Mendel's genetics, that is independent assortment. They are not dependent on anyone. They will be independent of their expression. Chromosomes from each homologous pairs are sorted randomly into pre-gametes. So this is the law of segregation given by Mendel. So law of segregation and law of independent assortment. Okay, so the chromosomes also follow this law while they are segregating, separating, and then coming together to form gametes of different varieties in the in the offspring to make the offspring. So they follow. They are more or less in line with the Mendel's law of inheritance. Parents synthesize gamete, and as we know, half of the parents, half comes from each of the parents. Half will come from each of the parents. This figure is representing meiosis, as you have studied in school also, the different stages of meiosis. So this is the part 1 of meiosis, and this is how you see the gametes which are formed. Okay, you can refer this. Nothing new. This is all you have studied in lower standard. Okay, and eggs and sperm have the same number of chromosomes. That is, they are gametes. They are haploids. And even the eggs will have half the number of chromosomes, the sperm will have half the number of chromosomes. Okay, and then they come together, they fertilize, they produce offspring with the same number of chromosomes as the parents. The offspring will also have the diploid condition. Okay, so these are some general understanding. You can say a recap or a revision of everything about chromosome and how they come together. What are the state chromosome level of the sperm and the egg, gametes basically, and the parents. These are some recap. Now we move on to the general understanding of the number of chromosomes. Okay, in a species, it will be specific and constant. If we are talking about, let's say, human species, in human we have 2 sets of chromosomes. In human, all humans will have 2 sets of chromosomes. That is what we meant by in a species, it is constant. So in humans, all humans, male, female, whoever it is across the globe, the diploidy level normal set of chromosome will be in every cell. It will be 2n. One n comes from the father, one n comes from the mother. And specific means, now here we are talking about human species. So it is 2n. In some other species, let's say in some honeybees, For example, it might be 3 sets of chromosomes. In some other organism, it might just be haploid. In honeybees, for example, sorry, haploid, monoploid in caterpillars. Okay, so that is also there. Depending upon species to species, they will have a particular set of chromosomes, and then it will be constant. Fine. And the term will begin with certain terms which are there on the screen. Ploidy means what? A degree of repetition of primary basic number. So this is the number of sets of chromosomes. For example, X in a cell. There can be two categories, euploidy and aneuploidy. First, we'll discuss about euploidy, which is divided into mono, di, and poly. Mono, di, and poly. Mono means after the set. Only one copy, one set of chromosomes. Diploidy, 2 sets. More than 2, 3, 4, 5. So poly. Poly stands for multiple. More than 2. So polyploidy. Okay, this is euploidy. Number of sets of chromosomes. Aneuploidy can be divided into hypo or hyper. Hypo means less than the required set of chromosome number. Hyper, more than the required set of chromosome number. So these are actually, this aneuploidy deals with the disorder state. Generally, aneuploidy is for disease condition or disorder condition. So there might be disease genetic disorders wherein 2n minus 1. Okay, 2n is a normal chromosome set that should be there in an individual, but one less chromosome is there in that person. So he or she might suffer from monosomy. Minus 2 sets of chromosomes. So minus 2 chromosomes, not 2 sets, 2 chromosomes. So the person might have nullisomy. Or one extra chromosome, that is also there. So this is subtraction and this is addition. But extra can result into this genetic disorder, trisomy. 2 extra chromosome, tetrasomy. So these are some of the genetic disorders which result into deletion of one chromosome or addition of one chromosome. So we are going to see examples regarding this in the last part of the chapter when we will discuss genetic disorders. Okay. So basically, this is a general understanding of the number of sets of chromosomes that should be present, and depending upon their copies, they have these different terminologies. Fine. Now we move to the structure, how the chromosome structure looks like. So this is how the structure of chromosome will look like. You are familiar with this, you have studied this in 8th standard, but still few more details will be required over here. Okay. So the first thing that we need to discuss is something known as centromere or kinetochore. Okay, centromere or another name is kinetochore. What is the centromere and kinetochore? You can see over here. Okay, this is the middle region, a constriction. Imagine Imagine there are 2 sticks and you tie it with a thread in the middle. Or let's say you have 2 4 sticks and you tie those bundle of sticks with a thread in the middle. So you are making a bundle of those sticks. In the middle, you're tying it with thread. So that tying up, you can imagine it as a centromere or a kinetochore. So primary constriction at the center, which chromatids or spindle fibers are attached. So from there, the spindle fibers or chromatids will come out or will be there. Spindle fibers are basically protein molecules which will arise at the time of cell division. Okay, so when the cell is dividing, so this is helping the movement of the chromosome during separation of this chromosome. So during cell division, you know these 2 parts they have to separate from one another. So that pulling away mechanism is brought about by the spindle fibers. Okay, this all you have studied in the lower standard. We are just doing a bit recap and few more details that needs to be added over here. So this is the centromere or the kinetochore. Another part of this chromosome is chromatid. So you can see these 2 parts, this part and this part. Okay, this part. So these are nothing but chromatids. So chromatids is each half of the chromosome joined. A chromatid is each half of the chromosome joined. They are again attached by this middle protein structure, centromere. This is your centromere. centromere. Okay, they are also known as sister chromatids. So two chromatids are there. So clear centromere, chromatid. Next is chromatid. What is chromatid? What is this chromatid made up of? This whole thing made up of chromosome, DNA, nucleotide. Okay, so condensed form. In the condensed form, we call it as chromatid. See, DNA, chromosome, chromatin, these are all the same thing. Chromosome is made up of DNA. When DNA is coiled and supercoiled together, okay, complex of DNA and proteins together, they form chromosome. And intermediate these structure which will result into formation of the sister chromatids, we will call it as chromatin within the nucleus of the eukaryotic cell. It is nothing but a combination of DNA and proteins, which is the chromosome only. Okay, so they are known as chromatin. Then in certain chromosome, in certain places, we will have the secondary construction also. If this is one of the chromatid, so you will find certain tying up construction over here also. This was the primary. This was a primary construction, and in certain chromosomes, you will see otherwhere also construction. Okay, somewhere away from the primary. So there we will call it as secondary. Generally, it is helpful in doing the nucleolar organization while the cell, after the cell has divided, and now is to reform the nuclear membrane and form a daughter cell. Okay. And telomere is the tip of the chromosome. Simple. It's the terminal region. It's the tip terminal region. Okay, so telomere is nothing but the terminal region of the chromosome. And finally, you have the chromonemata. Chromonemata is a part of chromatin only. So here there will be spirally wound filamentous structures, okay, onto which these chromomeres are arranged. Basically, how the shape takes place. That is nothing but the chromonemata. I will show you in the next picture all these labelings in bit detail. So centromere, middle primary constriction, secondary constriction somewhere away from the primary constriction. Chromatid is nothing but these two parts of the chromosome. Okay, chromatin is the material, the actual material of the chromosome, which is a combination of DNA and proteins. Telomere is the end part of the chromosome, the terminal region. T for telomere, T for terminal region. And chromonemata is the spirally coiled thread-like filamentous structures which makes up the chromatin. Okay, see here is how it looks like. This is the chromosome. This is the centromere, primary constriction. Somewhere you will see secondary constriction also. Okay. This is the outer membrane covering pellicle, and inside is the matrix. See, these chromosome which are spirally, coiled. This is basically, this chromosome is nothing but the chromatin and the DNA, which is combination of DNA and protein. So at this level, inside level chromonemata, primary constriction here you can see. Okay. So then you can have more than one secondary constriction. So one you can see here, one you can see here. Okay, from the primary constriction, the centromere and the spindle fiber will arise. Now, based upon the centromere, the primary constriction, the chromosome can be divided into 4 types. So, if the primary constriction is at the center, which is generally there, so metacentric, we'll call it as metacentric. If it is away from the center, either towards the top or towards the end, submeta. If it is near the ends, near the telomere, acrocentric. If it is at the tip, like this, then the chromosome will look like telocentric chromosome. So types of chromosome on the basis of position of the centromere. If it is in the center, metacentric. Slightly away from the center, either from towards this side or up or down, submeta. Near the telomeres, acro, and at the tip, if the centromere is at the tip, then telocentric. Okay, you should be knowing all these four names based on the position of kinetochore or the centromere. Okay, let's now go to the last part, that is the X and Y chromosome. Okay, so autosomes are all the same. 1 to 22 are the autosome, and then you have one pair of sex chromosomes. So out of that, one pair means two, na? So one is X and one is Y. Okay, as you can see in the picture, X is longer than Y. As you can see in the diagram over here, X is longer than Y, and Y is shorter than X. X contains more amount of euchromatin. Euchromatin means what? Lightly stained regions. Lightly stained regions. Okay, this we have discussed in the previous topic, in the previous chapter, and you might have also studied in lower standard also. Euchromatin means what? Under the microscope, they will appear light in color, lightly stained, because here the genes are very active and they are loosely arranged. They are loosely arranged. So euchromatin regions, okay, lightly stained. And what is heterochromatin? And in X, more amount of euchromatin is there and small amount of heterochromatin. Heterochromatin means where the genes, where the DNA is very compactly arranged. By the diagram, you can make it out. By the diagram, you can make it out. Where the DNA or the genetic material is loosely arranged. So here they will appear more light under the microscope, euchromatin, and heterochromatin. They are very compactly arranged. Okay, they are just coiled one top of the another. So they will appear darker under the microscope. Okay, so in X, this is more, this is less. In Y, it is opposite. In Y, more amount of heterochromatin and less amount of euchromatin. Okay, another explanation or another understanding you can have is euchromatins are having more active genes and heterochromatin are having inactive genes. Heterochromatin, active genes, inactive genes, and euchromatin, active genes. Okay, that is why they are loosely arranged. Then, so in X, more of U, less of hetero. In Y, more of hetero, less of U. Okay, you can see it over here. Next point, non-homologous region of X contains more genes. Now, we just saw what is homologous and non-homologous. Non-homologous pair, no similarity. Homologous means similarity. So this region we can see, they are having a similar shade of blue. So they have some similarity between them, between the X and Y. That is why they are known as homologous region. This is having no similarity, so non-homologous region. So non-homologous region of X have more genes. This is all of this is non-homologous region. They have more genes. Whereas in Y, non-homologous region has less genes. Because this is very short, Y is any which way shorter, non-homologous region is this much. So it is having very less genes. X-linked genes are present on non-homologous region. So whatever, so this is X and this is Y. So you have certain traits which come from X chromosome only. You have certain traits, certain characteristics which come from Y chromosome only. Also, the sex of the baby is determined by the presence of X chromosome or Y chromosome. So if it is 2X, then it will be a female. If it is 1X and 1Y, then it will be a male. Okay, this also specifies the gender depending upon what combination of chromosome is there. But the X-linked genes, most of the genes which come from the X, which give certain traits that we will see in the later part of the chapter, is there on the non-homologous region of the X, and Y-linked genes are present on non-homologous region of the Y. So that this is the example that we are going to see in the later part, like X-linked diseases, hemophilia, color blindness, night blindness, muscular dystrophy. Okay, these are some of the example out of which we will see night blindness and color blindness and hemophilia. Then Y-linked diseases can be hypotrichosis of the pinna or testicular atrophy. Okay, we are going to see this when we do a sex-linked inheritance. So this is a general understanding of Y and X. You should be knowing X is longer than Y. Then you have small amount of euchromatin in Y and more amount of heterochromatin in Y. More amount of euchromatin in X and less amount of heterochromatin. What is euchromatin? Active genes, loosely arranged DNA, loosely arranged DNA, loosely coiled, and heterochromatin is compactly arranged DNA, inactive genes. Mostly inactive genes are there over here, which are not used on regular basis. Okay. Non-homologous locus regions of X has more gene as compared to that of Y. Y has very less genes. So, and these are some of the examples of the X-linked diseases. The genes which are present on the non-homologous of the X chromosome, and then Y-linked disease are yours. So the genes which are present on the non-homologous regions of Y. So that's all. Make a note of it. If you have any difficulty, we will discuss definitely in the live class. So that's all. In the next video, we will see genetic recombination, independent assortment, and crossing over. These are basic concepts which you have studied in earlier standards also, lower standards also. So still we will go through this, because then further linkage concept is going to come. For that, we need to revise genetic recombination, independent assortment, and what is crossing over. So thank you. Till then, take care, keep revising, and see you. Bye bye.